Hello everyone, welcome to the Bodhi Academy. In today's video, we are going to continue with 12th Standard Biology Chapter 11, that is Biotechnology Principles and Processes. So in the last video, we have learnt about the cloning vectors. So we have learnt about the cloning vectors. What are the different features which are essential for cloning vectors, such as origin of replication, then selectable markers, cloning sites or the recognition sites, and the antibiotic resistance gene. Then we have also talked about the competent host for transformation of recombinant DNA. In this we talked about the microinjections, the biolistics or gene gun, and also about the disarmed pathogen vectors which are used. So today we will start with the processes of recombinant DNA technology. So let us begin. Processes of recombinant DNA technology. The first step in this process of recombinant DNA technology is the isolation of genetic material, that is the DNA. Now first, the DNA is enclosed in a membrane, right? It is enclosed, it is present inside the nucleus of the cell and it is enclosed by a membrane. Therefore, it is important to break the cell open in order to release the DNA along with several macromolecules which are present in the cell, such as RNA, proteins, lipids and polysaccharides. Now the isolation of DNA, this process is carried out in the presence of several enzymes like cellulase, which will be used for the plant cells, then lysozyme, which are used for the bacterial cell, and chitinase enzyme, which are used in case of fungus. Now different macromolecules are removed or isolated using different enzymes. For example, RNA is removed by treating with ribonuclease, whereas protein is removed by the treatment of protease, and other molecules are removed by other appropriate treatments. Now when these isolated or purified DNA is when chilled ethanol is added to it, the purified DNA is precipitated out as the collection of fine threads. So after this step, when the chilled ethanol is added to the DNA, it precipitates as a collection of fine threads in the suspension. So as we can see here, this is how the DNA will separate. Then talking about the cutting of DNA at specific location. So the next step is cutting of DNA using the restriction enzymes. So restriction enzyme digestions are carried out by incubating the purified DNA molecule with the restriction enzyme at suitable conditions. Then after this, the progression of restriction enzyme digestion, that is the cutting, is checked by the agarose gel electrophoresis. This is the process which helps us to check the or separate the DNA fragments which are cut from smaller strands to larger strands. Now since DNA is negatively charged, it is attracted towards the positive electrode, and this entire process is also repeated with the vector DNA as well. The DNA fragments after gel electrophoresis can be seen as bright orange colored bands when they are stained with the ethidium bromide stain and exposed to the UV light. So about this we have already learned in the previous videos. Then after elution, the cut out gene of interest, whatever gene that we have cut out, and also the cut vector are mixed together and the ligase enzyme is added. And as we have learned in the previous videos, ligase is the enzyme which is used to link or join different DNA fragments. So ligase is added, which then creates the recombinant DNA or the rDNA. Okay. Now this is the second step, that is cutting of DNA at specific location. Then the third step is the amplification of gene of interest using the polymerase chain reaction sorry, or the PCR. So we have already seen this isolation of genetic material, then cutting of DNA. Now the third step is amplification of gene of interest using PCR. So what is PCR? PCR stands for the polymerase chain reaction. So polymerase chain reaction is the synthesis of multiple copies. Okay, now we have already got a recombinant DNA. Now we want to make multiple copies of it, that is multiple copies of gene of interest in vitro. And what is used in this process? We need two sets of primers. Okay, we need two sets of primers and the enzyme called the DNA polymerase. Now we need two sets of primers. What are these primers? These primers are small chemically synthesized oligonucleotides that are complementary to the region of DNA. Now let us see the steps of PCR or the steps of polymerase chain reaction. So the first step here is the denaturation. First step is denaturation. So what happens in this step is, it is the heating of target DNA, that is the gene of interest, at high temperature at about 94 degree Celsius. So this is degree Celsius, to separate the strands, and each strand then acts as a template for DNA synthesis. So here we can see first is the denaturation. So first we have the double stranded DNA, then we heat the DNA at high temperatures. So the denaturation occurs, that is this strand and this strand separates. As you can see, this is one strand and this is the second strand. So the two strands are separating during denaturation. Then the next step is annealing. So what is annealing? It is the joining of two primers at 52 degrees Celsius. At 52 degrees Celsius at the 3' end of the DNA templates. Now after denaturation, both the single strand will act as templates. So what will happen during annealing? The two primers, now these are the two primers, this is first primer, this is the second primer, they will join at the 3' end. This is the 3' end, they will join over here, and this is the 3' end of the second one, and it will join over here. So they will join to the DNA template. Then this is the second step. Then the third step is the extension. So what happens during extension is that it is the addition of nucleotides to the primer using a thermostable DNA polymerase. So DNA polymerase is an enzyme, and the one which is used for this reaction is the Taq polymerase enzyme, which is a DNA polymerase. So it is the addition of nucleotides to the primer using a thermostable. Thermostable means this enzyme is stable at high temperatures. So as we can see here, to these strands the DNA polymerase, that is Taq polymerase, is added, and deoxynucleotides are also added, and then the extension will take place. See, so the Taq polymerase, that is DNA polymerase, is isolated. The enzyme is isolated from a bacterium called as Thermus aquaticus, and it remains active at high temperatures during the denaturation of the double strands. Now after the extension, what will happen? Through continuous replication, the DNA segment is amplified up to 1 billion copies. So now since the primers have attached and we have provided with the DNA polymerase and deoxynucleotides, the strand will continue to replicate. Okay, so this is how this single strand will it will again become double stranded. Okay, like this. And many copies will be formed, that is about up to billion times it will be amplified. Now the amplified fragment can be used later to ligate with the vector for further cloning. So it can be ligated using the DNA ligase to further clone to the vector for further cloning. So this is all in this video. Hope you have understood it. Thank you for watching my video. Please like, share and subscribe to the Bodhi Academy.